Good morning, everyone. My name is Hemlata and I'm your teacher. And we were discussing class 11 biotechnology, and this is our new chapter. We are going to start with this class 11 biotechnology chapter. I'm sharing the screen with you. So previously we have done with bioenergetics and enzymes, and now this is genetic disorders. This is chapter 8. So chromosomal abnormalities and the chromosomal disorders also come in the genetic disorders, the syndromes. In certain situations, due to environmental radiations, food intake, or internal genetic conditions, chromosomes may suffer damage. So, due to environmental radiation and the food we take and genetic conditions, chromosomes suffer damage and may change in number. The change in structure is called structural chromosomal abnormalities or aberration. This change in structure is known as structural chromosomal abnormalities or aberrations, and the change in number is called numerical chromosomal abnormalities. Structural chromosomal abnormalities are called aberrations. This is the difference, that structural chromosomal abnormality is called aberration, and the numerical is called the number change is called numerical chromosomal abnormality. So this we have to remind ourselves. When one chromosome of pair is absent, if one chromosome of the pair, out of the pair, if one chromosome is absent, it's called monosomy, 2n minus 1 for that chromosome. Monosomy of chromosome 1. When a chromosome is present in 3, means one chromosome is more. It's called trisomy, 2n plus 1. So, trisomy of chromosome X. It is important to notice that both monosomy and trisomy come under broad category of aneuploidy. So, both the monosomy means one chromosome is less and the trisomy, one chromosome more, come in the aneuploidy. However, when the entire set of chromosomes is multiplied, like 69, 23 into 3, if it is multiplied, and if it is, then this condition is called polyploid. It's not called aneuploid, it's called polyploid. Artificial breeding of plant has resulted in several polyploid varieties that we commonly use in our food. For example, bread, wheat has 6 sets of chromosome hexaploid. So wheat and bread, bread wheat, which is used for bread, is having hexaploid. Cabbage and mustards are tetraploids. Likewise, banana and apples are triploid, 3 sets of chromosomes. Strawberry and sugarcane are octaploid, 8 sets of chromosomes. Both the structural and the numerical changes can result in significant changes in phenotypic condition in the form of diseases or syndromes. Structural chromosomal abnormalities. Structural chromosomal abnormality may be following types. Deletion. In deletion, a segment of chromosome break away, leading to the shortening of chromosome. For example, retinoblastoma is caused due to the deletion of portion of chromosome 13. Sometimes when 2 ends of chromosomes are deleted, they can reattach to form a ring chromosome. So deletion, retinoblastoma is caused by the deletion. Duplication refers to when a segment of the chromosome gets repeated, resulting in a longer chromosome. This can lead to condition Charcot-Marie-Tooth disease caused due to the duplication of gene chromosome number 17. Inversion. Inversion, a segment of chromosome breaks away completely, reverses itself and reattaches with the chromosome. Here, the overall length of the chromosome remains same, but the orientation of the gene is reversed by 180 degrees. So inversion is the reversion of the chromosome. 180-degree reversion is seen in chromosome, and for example, RCAD syndrome caused by the inversion of the segment of chromosome 7, chromosome number 7, has reversed or inverted, so it's called inversion. And in chromosome number 17, not 7, 17 is being inverted in RCAD syndrome. Translocation. In translocation, a segment of chromosome breaks away and reattaches itself with another chromosome. एक क्रोमोसोम का कुछ पार्ट ब्रेक होके दूसरे क्रोमोसोम पे अगर लग जाए तो उसको ट्रांसलोकेशन कहते हैं. सो डिलीशन में सिंपली डिलीट हो जाएगा क्रोमोसोम का पार्ट, और डुप्लीकेशन में डबल हो जाएगा क्रोमोसोम का पार्ट. डायग्राम में आप देख रहे हो यहाँ पर बी पार्ट डिलीट हो गया है, शॉर्ट हो गया है क्रोमोसोम. डी पार्ट डबल हो गया है यहाँ पर, तो बढ़ गया है क्रोमोसोम, और इन्वर्जन में रिवर्स हो जाएगा क्रोमोसोम. ओके, सी इज showing the inversion of the B and the E part. The E part has come above and the B part has gone down. So E and B are 180-degree inverted. So B has changed into E. E has gone there at the place of B, and B has come at the place of downward. So it is inversion. And translocation is having this part, PQ part has come from another chromosome and translocated on A, B, C, D, E, F chromosome. So from here, it has gone to the another chromosome, this PQ, and has added to this chromosome. So this is the example of translocation. Fourth, so by all four diagrams, we understood deletion occurring in the first example, and duplication occurring in the second, and inversion occurring in this, and translocation occurring in this. So with this, we have understood this type of the shift in the chromosome. Reciprocal translocation becomes Burkitt's lymphoma, where exchange of material happens between chromosome 8 and 14. If a segment of chromosome breaks away and attaches with another chromosome without mutual exchange, it's called Robertsonian translocation. So if the mutual exchange is not there, just one chromosome is going, then it is called Robertsonian translocation. And in Burkitt's lymphoma, exchange of material happen between 8 and 14. Even the 8th chromosome and the 14th chromosome, both the chromosomes have break and attaches to one another. So that is reciprocal translocation. Numerical chromosome abnormalities, what happens in this? Some commonly observed syndromes, diseases due to the numerical chromosomal aberrations are described in the following sections. The term syndrome is generally referred to a group of symptoms which consistently occur together and characterized by a set of associated symptoms. So group of symptoms, if are coming together and considered to by a set of associated symptoms, this condition is known as syndrome. A disease referred to abnormal physiological response to internal and external factors. Example, fever caused due to microbes. और डिजीज अलग होती है. डिजीज एबनॉर्मल फिजियोलॉजिकल रिस्पॉन्स होती है इंटरनल और एक्सटर्नल फैक्टर्स की, जो माइक्रोब्स के थ्रू होती है. डाउन सिंड्रोम इंसिडेंट्स अकर इन अप्रोक्सीमेटली 1 per 800 birth. So 800 में से एक इंसान को डाउन सिंड्रोम होता है. Chromosomal basis है इसका. Down syndrome is a genetic condition that arises due to the presence of an extra chromosome 21. Here chromosome 21 is repeated thrice, trisomy of 21. Instead of showing up twice in a normal individual, the karyotype of Down syndrome is represented 47XX plus 21 females and 47XY plus 21 males. So 121 chromosome is extra in it, and karyogram of an individual. In this karyogram, you can see that all the chromosome are in the pair, but 21 chromosome having triple 21 chromosome is having a triple, we can say one extra chromosome in the Down syndrome individual, and whereas in the normal individual it is having only two. So B is the normal individual karyogram, and A is the Down syndrome person. So here we can see that in 21 chromosome, it's shown by the red that it is having 3 chromosomes. So 21 chromosome is having a nucleoide. One more chromosome is trisomy of 21, it's called. The trisomic condition is usually caused by an error in the process of cell division called disjunction. That is inability of the chromosome to separate at the time of cell division. So if the chromosome is unable to separate during the time of cell division, then this is known as non-disjunction. The trisomic condition is caused due to the non-disjunction of the error in the cell division. The possibility of having a Down syndrome baby in the family increases with the maternal age. If the mother is over 35 years, then the chances of having a Down syndrome child, that more than 85% Down syndrome babies are born in the mothers with over 35 years of age. At the time of pregnancy. Clinical symptoms. Some of the distinguishing features of the Down syndrome are flat face, slanting eyes, mouth, protruding tongue, single deep creases across the palm, low IQ, stunted growth, muscular hypotonia, undergrowth, underdevelopment gonads. Down syndrome babies also show breathing, heart, or hearing problem. Diagnosis and treatment. Down syndromes are usually diagnosed by an extra chromosome 21 in karyotype. There is no single standard treatment protocol for Down syndrome. Treatment are tailored on specific set of conditions presented by these individuals. At early age, children with Down syndrome can benefit from speech therapy, physiotherapy, and taking nutritional supplements. In early 1900s, on an average, Down syndrome age Down syndrome used to live until the age 9. Now, with the advances in the diagnostic and treatment technology, the age of expectancy has increased up to 60 and even longer. Klinefelter syndrome incidents occur in approximately 1 out of 1,000 newborn babies. Chromosomal basis, genotype 47 XXY affect males. The extra chromosome is not transmitted genetically, that is, Klinefelter newborns cannot have a Klinefelter father, but arises from inability of X chromosome to detach itself from the pair during meiosis. At the time of gamete formation, fertilization of an XX ova with a Y sperm produces an XXY zygote. The symptoms of Klinefelter syndrome are usually tall at their age, has reduced facial and body hair, smaller testes, and large breast and coarse voices. Diagnosis and treatment. One of the most frequent methods to diagnose Klinefelter syndrome is through bar body test of buccal smear. A normal no-bar body appears in the male buccal smear. However, in Klinefelter, one of the bar bodies show up, indicating the presence of an extra X chromosome. At the time of birth, babies with Klinefelter differ a little with the other normal babies. However, as their age increases, the differences between noticeable, especially at the time of puberty. People with Klinefelter syndromes are often treated with testosterone to look masculine. They also need to be psychologically counseled to control depression leading to aggression. Turner syndrome incidents occur in 1 in 2,500 newborn girls, frequently observed in miscarriages and stillbirths. Chromosomal basis affects females arises due to the missing X chromosome in affected females. This is called monosomy X, and karyotype is presented as 45X. A cell division occurs during the meiosis of an ovum, result in an ovum with no X chromosome and other with two X chromosome. The ovum with no X chromosome fuses with the sperm with one X chromosome to generate 45X condition. Mother with Turner syndrome cannot pass the condition to their daughter. That is, this condition is not inherited. The clinical symptoms, Turner syndromes are diagnosed by the following features. Short-stated, webbed neck, that is, neck skin is unusually loose and can be pulled several centimeters of the neck. Small breast, low set ears, that is, ears are placed below the normal position, swollen hands and feet. Furthermore, ovaries are underdeveloped and menstrual periods are usually absent. Diagnosis and treatment. Diagnosis usually happens through amniocentesis or chronic villus sampling. At puberty, the first test is that of bar body of buccal smear. The absence of bar body is the first indicator to follow up the condition with a detailed investigation. As with other syndromes, there is no permanent cure. However, growth and ovarian functions can be strengthened by the controlled administration of the hormones and androgen and testosterone and estrogen. People with syndromes. Famous people with syndromes. Isabel Zwingli is a famous fashion designer with Down syndrome. Isabel Zwingli. This feat wasn't easy as several universities rejected her application to study fashion. So, this is not easy to achieve because several universities have rejected her application to study fashion designing, but she persisted, and now the 19-year immensely talented person has showcased her work in London, Rome, and Mexico. It's commonly believed that the famous US President 6 feet 2 inches tall, Mr. George Washington, has Klinefelter syndrome. He has no children and also adopted later in his life. He adopted two in later his life. Laurel Foster, a Southern African model, was born male and diagnosed with Klinefelter condition. However, Lauren chose to identify herself with feminine features and transition into full female phenotype in her teens. Lauren became a successful model and appeared in Vogue magazine. She even wanted to contest for the Miss South African pageant but was disqualified. Hollywood TV, film, and stage actress Linda Hunt was diagnosed with the Turner syndrome. Linda started her career as a singer and made her Hollywood debut with Popeye film version. Linda has won 13 awards that include 2012 Teen Choice Award and 1984 Oscar Awards for the Best Supporting Actress. Dr. Catherine Ward Melber is a famous medical genetics doctor in the US, the 4 feet and 8 inches tall Melver was diagnosed with Turner syndrome. She was 7 years of age. Dr. Melver adopted 4-year-old Turner syndrome girl, Zoe from China. Monozygote genetic disorders and pedigree mapping. Monogenetic disease is caused by an error in the single gene. According to current estimate, 10,000 so 10,000 of the millions of the individual worldwide estimated to be monogenic. The nature of the disease, signs and symptoms depend on the function performed by the modified or defective gene. These diseases are inherited, and monogenic or Mendelian disorders, according to Mendel's law, they are inherited. In some cases, the mutation can be spontaneous, and other where we will not get the previous family history. So mutations in some cases are spontaneous. Here, family history is not present. There may be single mutation in the one gene and causing the specific disease like sickle cell anemia. So single gene mutation causes sickle cell anemia, or multiple type of mutation in one gene and producing the same diseases like cystic fibrosis. More than 200 different types of mutation can occur in one gene. The single gene or monogenic diseases can be classified in to the following categories according to the inheritance pattern as follows. So सिंगल जीन मोनोजेनिक डिजीज को हम नीचे दिए गए 4 पैटर्न में डाल सकते हैं. ऑटोसोमल रिसेसिव हो सकती हैं, ऑटोसोमल डोमिनेंट, एक्स लिंक्ड रिसेसिव, एक्स लिंक्ड डोमिनेंट. एंड यहाँ पर पेडिग्री के कुछ सिंबल्स दिए हुए हैं, वो हमें पता होने चाहिए. स्क्वायर रिप्रेजेंट मेल, सर्कल रिप्रेजेंट फीमेल, और अगर उनमें शेड कर रखा है, हमने पूरे फिल कर रखे हैं, तो ये अफेक्टेड इंडिविजुअल है. अगर हमने एक स्क्वायर को एक सर्कल से, एक लाइन से कनेक्ट कर रखा है, दैट मीन्स दे आर मैरिड पार्टनर्स हैं. ये स्क्वायर और सर्कल को अगर हमने लाइन से कर रखा है, और अगर हमने इस लाइन के नीचे एक और लाइन से दो अगर सर्कल और स्क्वायर लगाया, दैट मीन्स दीज आर ऑफ स्प्रिंग्स. सो लाइन के वर्टिकली डाउनवर्ड जब भी हम किसी सर्कल और स्क्वायर को लिखेंगे, दैट मीन्स ये ऑफ स्प्रिंग्स हैं. And from in order, the first offspring and then the second offspring. This will be in the order. And now this identical twins are given the next, and these are non-identical twins. If they are not connected by the lines, then they are non-identical twins. They are not separated by vertical line, they're separated by triangle. So these are the symbols which we use. Autosomal recessive disorder. The word recessive indicates that the 2 copies of the genes are needed to have the trait, and disorder in the case of mutated gene. Out of the 2 copies, 1 gene is inherited from father and 1 from the mother. So recessive indicate 2 copies are required for the defect to be expressed. And if an individual carries 1 defective recessive and 1 normal recessive gene, then he or she will be carried and not develop the disease. So if only 1 gene is defective, then that the person will become the carrier and not the affected by the disease on the basis of statistical projection. Statistical projection के basis पे estimated that every human being carry about 5 or more defective recessive genes that can cause a disease. Statistics से हमें ये पता चला है कि 5 ह्यूमन बीइंग में, हमें हर ह्यूमन बीइंग में 5 ऐसी डिफेक्टिव रिसेसिव जीन्स होती हैं जिससे कि उसे कोई बीमारी तो नहीं होती, वो उनका कैरियर होता है. The disease's phenotype of a recessive disorder is due to homozygosity of recessive alleles, and the unaffected phenotype is secondary to corresponding dominant allele. This can be explained with the example of sickle cell anemia, which is an autosomal recessive disease. Sickle cell disease is caused by a mutation in the hemoglobin beta gene found on the chromosome 11. This results in a defective hemoglobin Hb. After giving up oxygen, these defective Hb molecules cluster together, resulting in the formation of rod-like structures. The red blood cells become stiff and assume sickle cell shape. Red blood cells sickle cell anemia के sickle shape हो जाते हैं. So you can see the sickle cell shape red blood cells, which are very pointed at the ends, and they even get block. They even block the capillaries, and they do not transport the oxygen, just like the biconcave RBCs. And sickle cell anemia is determined by an allele which we designate as a S, and the normal condition by capital S. And the person affected by the disease will have a genotype small s, means both the recessive alleles will be inherited, and unaffected one will be having capital S, or heterozygous. We can make a projected pedigree of the disease as shown in the figure, assuming that both the parents are carriers. Sickle cell anemia is particularly common among people whose ancestor originated in sub-Saharan Africa, South America, Cuba, Central America, Saudi Arabia, India, Mediterranean countries. In India, it's common among people of Deccan Plateau of Central India, with a smaller focus in north of the Kerala and Tamil Nadu. For example, of autosomal recessive disease include other examples are autosomal recessive disease, cystic fibrosis, Tay-Sachs disease, phenylketonuria. Individual with the cystic fibrosis produce mucus that is normally thick and sticky that can damage different organs, especially lungs, resulting in chronic infection. Tay-Sachs disease is due to the absence of enzyme called hexosaminidase, which result in the fatty substance accumulation in nerve cell, particularly affecting the brain. It is fatal disease manifested in childhood. 1 in 27% of Europe, Ashkenazi Jewish origin individual carries Tay-Sachs gene, and phenylketonuria is caused by mutation in phenylalanine hydroxylase gene, resulting in the increase in the phenylalanine in the blood. Normal red blood cells, sickle cell trait, and sickle cell disease inheritance is shown by this cross here. Normal, they are heterozygous, both the individual, and the chances of having a sickle cell trait in their offspring is 25%. Autosomal dominant disorder, achondroplasia causing dwarfism, and then X-linked recessive disorders are shown. So we'll be doing autosomal dominant disorders, X-linked recessive disorders, and some X-linked dominant disorders in our next video. Polygenic disorders also we will be covering in our next video, because today we have discussed about the introduction of the genetic disorder chapter, and the types of disorders, and even the types of chromosomal disorders we have discussed in this chapter. So with this, for preventing the video to become very long, so I would like to end the topic here. We have discussed the Down syndrome and the chromosomal abnormalities and the syndromes. So, and one of the monogenic disorders we have discussed, and some introduction of the pedigree we have discussed. So monogenetic disorder में हमने sickle cell anemia discuss कर लिया और उसकी inheritance समझ ली. अब हम X-linked recessive disorders and dominant disorders को next video में समझेंगे. Till then, bye bye. Thank you for watching. If you like the video, then like the like button, press the like button, and share and subscribe my channel for such more videos of class 11 biotechnology. Thank you.